Introduction to Stem Cell Research. Stem cell research is the area of research that studies the properties of stem cells and their potential use in medicine. As stem cells are the source of all tissues, understanding their properties helps in our understanding of the healthy and diseased body's development and homeostasis. How does stem cell research work? Researchers grow stem cells in a lab. These stem cells are manipulated to specialize into specific type of cells, such as heart muscle cells, blood cells, or nerve cells. Then the specialized cells can then be implanted into a person. Why is there such an interest in stem cells? First, researchers and doctors hope stem cell studies can help to increase understanding of how diseases occur. By watching stem cells mature into cells in bones, heart muscle, nerves, and other organs and tissue, researchers and doctors may better understand how diseases and conditions develop. Second, generate healthy cells to replace diseased cells. Stem cells can be guided into becoming specific cells that can be used to regenerate and repair diseased or damaged tissues in people. Lastly, test new drugs for safety and effectiveness. Before using investigational drugs in people, researchers can use some types of stem cells to test the drugs for safety and quality. These are some of the pros and cons of stem cell research.
Stem cell therapy is a brand new type of technology that aims to replace damaged cells with healthier ones, though there are still multiple challenges that it faces before it can be distributed to the masses. Here are its 5 main challenges. Stem cell therapy has a wide array of processes that must be considered prior to administration. The multiple factors and components prove to be rather complicated, though they must be concise. Thus, this aspect of stem cell therapy is still in progress of being assessed, tested, and approved. Until it can be approved, issues with manufacturing prove to be a challenge. The inherent genetic instabilities of stem cells, specifically in HESCs and iPSCs, pose a challenge to the regulation of stem cell therapy. This is why it is important to both perform detailed analysis such as chromosomal aberration and establish degrees of accepted genetic change. The manufactured cell must be fully characterized, predicted, and free from contamination. However, as the passage number of stem cell increases, the risk of chromosomal irregularities also rises. This is why tests and procedures are done in hopes to minimize the number of passage lines, controlling the chance of genetic variation. Stem cell therapy cannot be held to the same standards as other drugs or biopharmaceuticals. Stem cells are highly sensitive to drugs, with issues such as transplant rejection from post-operational care. This is why scientists intend on studying the pharmacokinetic behavior of stem cells by analyzing its effects on a larger animal model. Despite the multiple challenges, there are promising solutions which are sure to progress humanity in the field of health and science.
Researchers working on stem cell therapies worry that stem cells would grow in unexpected ways or situations, establishing the wrong tissue in the wrong place. In this report, we're going to tackle the side effects of using stem cell therapy. In 2016, a 77-year-old woman went to a clinic to have stem cells injected in her eyes. Within 3 months, her retinas had peeled away from the rest of her eyes. As a result, she can only make out hand motions in her right eye and could no longer walk on her own. A 64-year-old patient who underwent stem cell therapy for his backbone pain was paralyzed because of injecting the fat stem cells and approved by the FDA. Hence, the effectiveness of stem cells as a primary remedy option in spinal cord injury is currently under much examination. There are also other possible side effects of stem cell therapy that might happen in the long run, such as the following. There are a few deceitful providers who offer stem cell products that are neither proven nor approved. So, we must always be careful about the potential risks involved in this procedure and check what is being provided to us in the name of treatments, because no one knows what will happen when experimental therapy is tried in humans.
Sometimes, a part of a gene is defective or missing from birth, or may change during adult life. Any of these variations can disrupt how proteins are made, which can contribute to health problems. Gene therapy is the proposed solution to the problem the process by which scientists attempt to correct certain genetic abnormalities and the terminal conditions caused by these genetic issues, treat and prevent diseases through the insertion of genes into a patient's cell instead of using drug or surgery. They can either replace a gene that causes a medical problem with one that doesn't, add gene to help the body to fight or treat disease, or turn off genes that are causing problems. Gene insertion is the only possible version of gene therapy in use today. With gene insertion, genetic material is inserted into the patient via any number of vectors. If this insertion is successful, it becomes a fully functional protein. This can cause the targeted cells to produce additional types of proteins which can have impact of reversing the condition of certain diseases. In order to insert new genes directly into cells, scientists use a vehicle called a vector, which is genetically engineered to deliver the gene. Truly, gene therapy brought hope to humanity in curing the incurable diseases such as cancer, and brought faith to many in surviving their problems in life.
Challenges of gene therapy. Although several protocols have been successful, the gene therapy process remains complex, and many techniques need new developments. The specific body cells that need treatment should be identified and accessible. A way to effectively distribute the gene copies to the cells must be available, and the diseases and their strict genetic bonds need to be completely understood. In gene therapy, a normal gene is inserted into the genome to replace an abnormal gene responsible for causing a certain disease. Of the various challenges involved in the process, one of the most significant is the difficulty in releasing the gene into the stem cell. Thus, a molecular carrier called a vector is used to release the gene, which needs to be very specific. Once the vector is inserted into the patient, it cannot induce allergic reactions or inflammatory process. It should increase the normal functions, correct deficiencies, or inhibit deleterious activities. Finally, the vector should be capable to express the gene. Furthermore, it should be safe not only for the patient, but also for the environment and for the professionals who manipulate it. In general, for the patient's entire life.
Gene therapy has some potential risk. A gene can't easily be inserted directly into your cells. Rather, it usually has to be delivered using a carrier called a vector. This technique presents the following risks. Firstly, unwanted immune system reaction. Your body's immune system may see the newly introduced viruses as intruders and attack them. This may cause inflammation and in severe cases, organ failure. Secondly, targeting the wrong cells. Because viruses can affect more than one type of cells, it is possible that the altered viruses may infect additional cells, not just the targeted cells containing mutated genes. If this happens, healthy cells may be damaged, causing other illness or diseases such as cancer. Thirdly, infection caused by the virus. It is possible that once introduced into the body, the viruses may recover their original ability to cause disease. Fourthly, possibility of causing a tumor. If the new genes get inserted in the wrong spot in your DNA, there is a chance that the insertion might lead to tumor formation. And lastly, gene therapy may result in too much of the protein being created. The effect of this overproduction or overexpression could vary based on the type of protein being created.